Well, greetings everyone and welcome to the National Institute on Aging Common Data Elements webinar. My name is Dr. Karen Anyanaho and I'm the advisor for genomic data sharing with the National Institute on Aging, or NIA, in the Division of Neuroscience. I am joined today by my colleague, Erin Mazam, who collaborated with me in spearheading today's lecture. Before we get started, I just wanted to go over a few housekeeping notes. One is that our audience is currently muted. In-meeting chat has been disabled. However, the Q&A window is enabled because we want to solicit your questions throughout the presentation. And you'll see that appearing during the Q&A segment of today's lecture. Cart captioning is also provided today by Ms. Adrena Atkins. And I also wanted to acknowledge our colleagues within the NIA providing IT services, Mr. Raul Maston and Mr. Dwayne Diggs. So today I'm very pleased to welcome you to our third monthly webinar series as a part of our Common Data Element Speaker Series, which most of you know began April, will run through December of 2024. We are featuring a dynamic lineup of speakers who will provide insights from their research findings relevant to how common data elements, or CDEs, are defined in both human and machine computable terms, as well as highlight best practices about standards and applications for optimizing data harmonization. Our speakers will also share insights about the usage of data resources, effective data management, and enhanced data analyses for the development and use of common data elements. So our speaker for today is Dr. Tim Holman. You'll hear about him in just a moment. This series is really intended to allow the NIA community to connect with Alzheimer's disease and Alzheimer's disease-related dementia and aging research, and with researchers who have expert knowledge in the field. We want to facilitate discussions about the resulting research findings, as well as provide a space to share valuable information as it relates to CDEs. So this afternoon, we are delighted to have Dr. Tim Holman as our featured speaker for today's webinar, which is titled Application of Computational Data Harmonization Approaches to Study the Structure and Function of Alzheimer's disease and Alzheimer's disease-related dementia, better known as AD-ADRD. Now, Dr. Holman is an associate professor of neurology, cognitive neuroscience, and computational genetics, and has secondary appointments in the Vanderbilt Genetics Institute and the Vanderbilt School of Medicine's Department of Pharmacology. Dr. Holman leverages advanced computational approaches from genomics, proteomics, and neuroscience to identify novel markers of Alzheimer's disease risk and resilience. He received his doctoral degree in neuroscience from American University, focusing on cognitive and neural changes during normal aging. Today, Dr. Holman will discuss computational infrastructures that support the processing of many harmonized Alzheimer's disease and Alzheimer's disease-related dementia data. And in this vein, he'll also discuss what is needed to support data harmonization. Further, Dr. Holman will highlight effective approaches towards studying the structure and function of AD-ADRD. So Dr. Holman, without further ado, I will now turn over the presentation to you. Thank you.
Great, thanks so much. I'm really excited to present to you guys today. Share my screen. All right, thanks for that great introduction, Karen, and this opportunity to talk about the Alzheimer's disease sequencing project's phenotype harmonization consortium. My disclosures: I was previously a scientific advisor for Vivid Genomics, and I'm a deputy editor for Alzheimer's and Dementia, TRCI, and a senior associate editor for Alzheimer's and Dementia. So, oops, sorry, I can see the top of my screen isn't working right. So I'll start that again. Oh, sorry. Looks like the top of the screen you're going to have a little overlap, but so maybe I'll just start by talking a little bit about the Alzheimer's disease sequencing project and the opportunity that we have as a field right now with a really unique data explosion that's happened in the last decade. So over the last now 12 years, the Alzheimer's disease sequencing project has been generating whole genome sequencing to study rare genetic variants that contribute to late onset Alzheimer's disease. And the study started with the goal of doing sequencing on 5,000 participants. And most recently, we released data from more than 36,000 participants that have whole genome sequencing data. And the goal by 2025 is to up that number to 100,000 participants that have whole genome sequencing data. And at the same time that this, really huge data generation activity has been happening in whole genome sequencing, there's also been a ton of GWAS data that's come out. So the largest Alzheimer's disease genetic study recently had over 1 million participants included in it that had genotype data. So this is a great time if you're a computational scientist like I am, to be able to study genetic factors that contribute to Alzheimer's disease. But what's really unique in the Alzheimer's disease field is at the same time that this genomic data has been expanding, a lot of the cohort studies that come into the Alzheimer's disease sequencing project also collect molecular biomarkers longitudinally. So now it's possible to measure in vivo the primary neuropathologies that contribute to Alzheimer's disease, amyloid beta plaques and neurofibrillary tangles. And we can measure those, we can measure biomarkers of those in life using proteomic techniques in blood and in cerebral spinal fluid. And we can also use PET imaging to look at where these neuropathologies are actually located in the brain of living individuals. At the same time that those molecular biomarkers have been expanding, there's also been an expansion of structural brain imaging techniques to look at how the neurodegenerative processes that occur with Alzheimer's disease and related dementias change over the course of disease as well. So this is another huge opportunity that a lot of the cohort studies that are included in these large genetic consortia have also been collecting. And then, of course, we also need to track people's cognition if we're going to look at the clinical manifestation of disease. A lot of the cohort studies also have longitudinal cognitive data that can be used. So in the last few years, the Alzheimer's disease sequencing project has started a few new initiatives to try and leverage this incredible opportunity with all of the genetic data that's being generated. So those three initiatives, one of them is a artificial intelligence and machine learning initiative with the goal of identifying multi-omic signatures of Alzheimer's disease and related dementias. A second is a functional genetics consortia called FunGen that's trying to move from here's the genetic factor that we have, what's the actual gene and mechanism that's underlying these variant level effects that we're seeing. And then a third is the initiative I'm going to spend all today talking about, which is the Alzheimer's disease sequencing project phenotype harmonization consortium. So that initiative, to expand on it a little bit, was funded to work in coordination with the other Alzheimer's disease sequencing project work groups and initiatives to streamline access to endophenotype data, provide high quality phenotype harmonization across a number of domains that I'll show you in just a few minutes, provide comprehensive documentation for how we're doing this data harmonization, and make both our harmonization procedures and code and also the harmonized data available to the research community. So we had 4 aims in this project: to procure, collate, curate endophenotype data from all the cohort studies that are in the ADSP, to harmonize those data, to disseminate those protocols and the harmonized phenotypes, and to educate the research community. So I'm going to tell you a little bit about some of the things that we've been doing in this space, and hopefully that'll generate some conversation that we can have around opportunities and challenges that we've seen in our work on common data elements and generating harmonized data. So first, I want to highlight the infrastructure that underlies this particular project. So the Alzheimer's disease sequencing project is a really large project that includes a lot of different grants and mechanisms, but all the data for the ADSP is shared in one central location, which is NIGAD. So NIGAD is the resource for genomic data sharing within the NIA. And the way that we set up this project, and really could only do this project because this infrastructure was already in place in NIGADs, is we reached out to the cohort studies that are included in the ADSP, and we asked if they'd be willing to share more deep longitudinal phenotype data as part of this genomic data sharing that they were already doing, so that our harmonization teams could access those data right on NIGADs, could curate, document, harmonize, and then put those data back up on NIGADs so that anyone who's already using ADSP data can now access a lot more information on the participants that have already been sequenced. So it's a huge value add to what's already a really valuable study. But the reason this is so important, and this workflow is a little bit complicated, is because without this infrastructure in place, we'd be required to go to 50 different studies and set up data use agreements with each one of those for everyone on our harmonization teams. And then everyone who accessed the data after that would have to do the same thing as well. So the infrastructure and time and energy that's been put in place by NIGADS, it's run by Dr. Lisan Wong at the University of Pennsylvania, made this project possible. So we're really grateful. We work very closely with those guys, and very grateful for all the time and energy that they've put into this incredible resource. So the way that works is, we reach out, of course, the National Alzheimer's Coordinating Center, NAC, which is housed at the University of Washington, plays a huge part in this. A lot of the participants that are in the ADSP come out of NAC, out of the Alzheimer's disease research centers around the country. But there's a lot of other cohort studies as well that are included that are not part of NAC. We reach out to those, we contact them, we talk about the project, we ask if they'd be willing to share phenotypes and images, and their genomic data. They set up a DTA with NIGADS, they share their data with NIGADS, our harmonization teams access those, harmonize, and then make them available. One of the challenges that we had right out of the gate was how we were going to handle neuroimaging data. I mentioned that there's a ton of neuroimaging data, and that's one of the really incredible opportunities that we have. But NIGADS isn't set up to do neuroimaging data sharing or DICOM sorting or any of those things. So we partnered with the Laboratory of Neuroimaging, led by Dr. Arthur Toga at USC. And they, in partnership with NIGADS, have created back-end handshake solutions to facilitate data sharing on LONI for neuroimaging data, while all the scalar data is shared and accessed directly on NIGADS, making that process pretty seamless for the user community. So really grateful for that partnership, and I'll talk a little bit more about that. But the big takeaway is everything I'm going to present to you today, you can go to dss.nigads.org and access all the data, and hopefully coming out of this, there'll be some ideas that you have for cool genetic studies that you could perform, and you can go on and do that today. So that's the infrastructure that underlies what we're doing. So here are the harmonization domains that are included in our projects and the domain leads for those different parts of the project. So first, we have 3 coordinating centers here at Vanderbilt. We are the primary data coordinating site. Dr. Mike Kukro at the University of Miami leads our integration with the other ADSP work groups and projects. And Dr. Arthur Toga, I mentioned at USC, is leading the image sharing and imaging infrastructure for the overall project. And then for each of these different domains that are listed out on this slide, we reach out and ask leaders in the field if they'd be willing to do the data harmonization for this project. So when we submitted the application, we only included domains for data harmonization that already had established techniques in the literature. And we leveraged those approaches. So you'll notice, one of the things that we didn't do, for example, is we didn't do blood-based biomarker harmonization for the emerging blood-based biomarkers of disease. And that's because there wasn't yet, and that's still an area of need, an agreed upon harmonization approach for that particular domain. So we didn't do it. But for these domains, cognitive data for vascular risk factors, neuropathology data measured at autopsy, PET imaging, structural MRI, diffusion MRI, and cerebral spinal fluid measures of Alzheimer's disease neuropathology. There's really good literature on harmonization approaches that we felt confident in. So those are the domains that we ended up choosing. And then I'll just say each of these domain leads puts an incredible amount of time and effort to make these data, to harmonize these data and to make their scripts and their code and their harmonized phenotypes available to the research community. So really grateful to have an incredible team that we get to work with on this project. We just had our investigator meeting here at Vanderbilt over the last 2 days. So had a lot of time spending time in person with these folks over the last 2 days as well, and really grateful for their partnership. The one other piece I want to highlight is this project on the Million Veterans program out of the VA that's being led by Dr. Mark Logue and Dr. Debbie Swam to try to integrate components from the EHR into the harmonization that we're doing here. And really as a proof of concept that we're able to do that. And I think will set the foundation for integration into the real world data types that, are a future direction of something that we certainly need to do. So I'm happy to talk more about that, but I'll just mention that right now that I'm not going to go into too much detail on the MVP coordination, but that's just getting off the ground and we're really excited about it. So what do we actually deliver in this project? So for each of the domains, we're thinking about two different users when we developed the deliverables for the project. One user is a geneticist who wants to leverage some of these deeper end of phenotypes that can tell you about how Alzheimer's disease is changing over the preclinical stages of disease. That can be 15 to 20 years. For the geneticists who may not have domain expertise, we make simplified phenotypes available that they can use very quickly and easily in their data analyses. So for example, from our amyloid PET analysis, we make a amyloid positive negative score available for participants who are in the ADSP. That can just be brought into any standard GWAS pipeline, for example. The second user that we're thinking about is the domain expert user that are also going to want access to these types of data. And for those, we release really granular data that can be leveraged to redo our harmonization if you don't like the approach that we took, or can be used to look at regional values, for example, for amyloid PET or change in amyloid PET within a given region. So we release not just scalar data up on NIGADs, we also release imaging data collections on LONI, so folks can go in and leverage some of their own pipelines and resources to do this amazing work. So happy to take questions on the specifics for any one of these, but I'll give you a little taste of maybe 3 or 4 domains in a few slides and talk through the approaches that we use statistically to do this challenging work of harmonizing across many different scanners, many different sites, many different cognitive batteries for how that approach actually works. So we had our first data release in 2023, and this was the data that was in that first release. Released data from 15,000 participants that had longitudinal cognitive data. For most of those studies, the average participant had about 5 years of follow-up. And yeah, so those data were made available 2 years ago. And then last year, we had promised for year 2 in our deliverable that we would deliver for these 4 cohort studies and these different domains. But what we actually were able to release was a lot more data than that. And this is just some of the numbers that we released in December of last year. So this is the overall harmonization across each of these domains. These are all the participants. These are participant numbers, not visits. All the participants that were included in our harmonization that was released last year. You can see we had cognitive data harmonized for more than 80,000 participants. We had images for more than 10,000 participants that were harmonized for this project. And you can see across the different cohorts how those numbers lay out. The way that the data release works, and for the project, this is obviously for the ADSP. So we do the data harmonization in the overall cohort because often the people who are selected for sequencing are a subset of the cohort. And we want to make sure that the harmonization is as robust as possible. So we actually ask the cohorts for all their data, even if their participant wasn't sequenced in ADSP. And then from that overall subset, these are the participants that were actually uploaded to NIGADs for use in the Alzheimer's disease sequencing project. So on NIGADs, on dss.nigads.org, you can log in and access data from these 25,000 participants that have whole genome sequencing and also have these phenotypes. So let me walk through some of the approaches that we actually use to do data harmonization to give you a sense of the work, energy, and effort that's gone into this project. And I can highlight some of the cool domain leads that we get the pleasure of working with every day. So I'll start with cognition. So across these different cohort studies, many of them use different neuropsych protocols to do data collection. And what we do for the cognitive harmonization is we reach out to each study and we ask for a lot of details from their neuropsych batteries. So we ask them to send us their forms that they use. And then we have a panel of experts that include 2 neuropsychologists and a neurologist, who will go through every item that's administered in that particular study, and they'll make a few decisions. The first decision they'll make is, for this given item, is the item measuring memory function or language function or executive function or visual spatial ability, and they'll categorize that item into a given domain. And the second thing that they do that's really important for our approach is they ask whether or not an item was given in an identical way across studies. So in this example, for memory performance, you can see here we have data from 4 different cohort studies that were included in our first data freeze 2 years ago. And a lot of items that you can see in individual colors here were given in one study, but weren't given in the other study. So those we can't use as anchor items. We're going to freely estimate those items. But a whole bunch of items were given across studies, and we can use those items that were administered consistently across studies to act as anchors so that our final composite score means the same thing in ROS Map Mars as it does in NAC. And that means that a 1-point increase, regardless of the study that you're in, means a 1-point increase. So it's a really cool approach that allows us to both anchor things that the score has the same meaning across studies, but also keeps us from having to throw out a ton of data that was collected in these different studies. That gives us more sensitivity to true memory performance in those studies. So after we do this, you end up with really nice, normally distributed cognitive measures that are longitudinal and can be leveraged in genomic analysis. And here's just an example for our most recent data release across studies. 2 of the scores that we had, memory score and our executive function score. And once we have those data, now we make those available. And actually, this study here came out, before my team had even published anything off of this. Another group in the ADSP PhD already had jumped in and ran an analysis and did a really cool GWAS where they looked at pleiotropy across these different cognitive domains and looked for genetic factors that relate to executive function, memory, language individually, and then also predict more than one of those, and found some really cool results in that. We also published the harmonization co-calibration approach. This was led by Joy Mukherjee at the University of Washington. And then we took the data and we asked, okay, what are some really basic but important questions that we can ask now that we have a data set of this size? And a great analyst on our team here at Vanderbilt, Skylar Walters, co-led this project with a PhD student here, Alex Contreras, led by an assistant professor on our team, Logan Dumitrescu. And they looked at whether the ApoE genotype was related to these different domains of cognition, whether that association differed by sex, and whether it differed across racial ethnic backgrounds, and was able to show first that the sex difference in E4 is consistently observed in relation to both language and memory function, but also that sex difference is present across racial groups as well. So that was one of the first studies to show that. So really just a cool application of what we can do when we have harmonized data at this sample size, and hopefully just the beginning of what's going to come as we continue to release more and more data. So as another example, we also have a really detailed protocol for doing harmonization of diffusion imaging. This is led by Bennett Landman here and Derek Archer here, who's this professor on our team. And what these guys do is they take all the raw images in. They have a really nice standard diffusion processing pipeline that takes care of artifacts and takes care of motion. Every scan from every study goes through the exact same protocol, has a PDF that prints out about summarizing the pre-processing that was done. And then what they do is, following that kind of pre-statistical harmonization where they're getting all the images to be on a similar sort of scale, they then do a statistical harmonization approach using longitudinal combat that takes care of other scanner effects that aren't taken care of in that first step. One of the things that's really unique in this particular project too, Dr. Derek Archer is an expert in free water imaging, so they also calculate a free water component and corrects the measures in white matter for that free water compartment. And all those data are made available as well. And again, that's the images if you want to access the images. but also the scalar data are made available too. And this is just one example of a project led by Dr. Archer, where first he just kind of demonstrated this harmonization works really well. But then once the data are harmonized, he can look at the effect of age across these cohorts on these different measures of white matter structure and function, and basically found that there's this really large effect of aging on a measure of free water in the cingulum bundle, and showed, for the first time at this scale that you have this really sensitive aging effect that we can pick out across disease status. So another cool example of some of the data that we're making available and some of the applications that are coming out of that as well. For amyloid and tau PET imaging, this is led by Dr. Doyu Tosun and Dr. Elizabeth Reno, who are both experts in PET imaging. A lot of the challenges that we have in MRI are present in PET as well. So for amyloid and tau PET, many of the studies use different ligands. Many of them are on different scanner platforms. So the problem that they've had to solve is how can we get all of these pass through consistent pipeline where we can be confident in the scores at the end and use them in joint analysis. So they have a pipeline that they developed based on a published protocol from Susan Landau. One of the things that's interesting is a lot of the PET images that come in without a MRI scan with them. So they developed an MRI-free approach to maximize our sample size and demonstrated a really robust harmonization approach that gives a few different measures that can be used in downstream analysis. One of those is a centiloid approach, which is a linear transformation that can be done to take any amyloid ligand and put it on a PIB scale, essentially, that's then scaled. And some of the studies were not able to calculate centiloids because of the way the data were captured or made available to us. And in that case, we use a statistical approach post-processing that is based on a Gaussian mixture model. And essentially, you can use the distribution of the data to do this really nice nonlinear transformation across ligands that gets everything back onto a same scale that can be jointly analyzed. So after all that's done, they then document it, and those deliverables are put up, both the harmonized metrics that can be used in analysis, but also the code and the approach so that people can apply those in their data sets if they so choose. And also, can take another approach if they don't prefer what we've got. So far, they've completed this harmonization on more than 9,000 amyloid PET scans and 3,000 tau PET scans. And these data are just continuing to grow. So we have quite a few other harmonization approaches. I won't go in quite as much detail on these ones, but for the fluid biomarker harmonization, we again use an approach that's based on Gaussian mixture models and clustering to walk across different proteomic platforms that are used to capture A beta and tau. For our neuropath harmonization, this is probably when I'm thinking about common data elements and a lot of the topics that have been discussed on this previously. This is really very similar to most of the common data element mapping approaches that are in the literature and that are used across a lot of other studies. What we did for this is we went to the neuropath forms. This is led by Dr. Tom Montine and Dr. Gary Beecham. We went to the neuropath forms from NAC, and we map every study's neuropath categories onto those NAC forms. And they make a lot of challenging decisions too. For some studies, you don't have the granularity that you have in the NAC neuropath form. So how can we map those on to a variable that's common across data sets that maybe has a little bit less granularity than what you have? But for the studies that do have really granular data, we can map it all the way to the NAC neuropath form. So these data are again made available right on NIGADS, and they've been incredibly productive and have these really nice coding maps where you can look for a given study, what's the crosswalk if you want to get to the NAC neuropath forms. And then for structural T1, our pipelines are very similar to what I was showing for diffusion, where we have pre-statistical harmonization that goes through and does image transformation. But in the structural T1, there's also a deep learning algorithm that's used to correct and adjust for nonlinear effects of age and account for batch and scanner effects as well. And this produces both a score for Alzheimer's disease risk that's can be applied pretty easily in any study, but also region of interest data across a whole bunch of parcellated regions from a few different atlases that can be used if someone has a region-specific hypothesis for how they want to look at neurodegeneration. So a few other examples of some of the work that's going on in the data that are being released. So just a few updates on what's coming down the road. And then at the end, I'll give a few other scientific examples. I couldn't help it. I'm a scientist first and foremost. Data harmonization I've really enjoyed doing, but certainly we harmonize data so that we can discover new scientific discoveries. So the updates that are coming. So first in the spring, what we're releasing now is these imaging collections. So we just finalized one of these collections that is going up to Loney, and now we're finalizing the rest of those to put up. This makes it so that the community, one of the big deliverables that we had in mind for the imaging collections in particular are the AI and machine learning work groups within the ADSP and elsewhere that are trying to benchmark approaches. And it's really important when you're doing this benchmarking that everyone's using the same input data and the same test data, and they have an out-of-sample data set that they can use as well. These collections have facilitated our ability to support some of those projects and make sure that everyone across the different teams are working from the same data. We also, in the fall, will be doing our Freeze 3, our third data release up on DSS. We expect that'll be November of this year. And here are some of the data sets that we're working on for the new cohorts. So you can see on the top row the work that we've done so far, and then on the second row are the cohorts that are coming in. So even though we've released a ton of data, we're really just scratching the surface. There's so much more work to do. There's so many additional data sets to bring in. And I think this resource is just going to continue to grow and continue to have an impact. So we're really excited to be bringing in a few data sets. Maybe I'll just highlight one or two of these. The HABS HD data set is a data set that's led by Dr. Sid O'Brien. And they have an incredible amount of diversity in that data set. And they've done imaging biomarkers on a lot of that data set too. So that includes more than 4,000 individuals. And it has a really high rate of both African-American participants and also Hispanic participants in that study. So really excited to see that. You can also see quite a few of these cohorts are coming from around the world. So it's not just US data sets that are coming in. There's a data set from Korea that's coming in. Quite a few data sets are being generated in Africa right now that are coming in. Quite a few data sets from South America are coming in. So we've been very blessed to have the opportunity to work with these different cohort studies and bring in data from around the world to answer important questions about the genetics of Alzheimer's disease. So I mentioned the MVP project. We had a competitive revision that was just recently funded to do this integration with the Million Veterans Program in the VA. We'll be excited to report back on that next year. And then the other thing that we've had these opportunities to grow what we're doing as we've gone through and started doing data harmonization. We've just seen opportunities to build on what's already present in the ADSP. Just as one example of that, a lot of the cohort studies, when they uploaded phenotypes to the ADSP originally, they would just give information about whether or not a participant was a case or a control, whether they were an Alzheimer's disease case or a control. But now that we have pulled in all the longitudinal cognitive data and the longitudinal data from imaging, a lot of those cohorts also have longitudinal diagnosis data. So we've established a work group to go through and adjudicate those different ways that diagnoses are recorded across data sets and integrate that into the phenotype information that we're making available. So you can not only look at an imaging feature longitudinally, but you can also know what that participant's diagnosis was at that time point and make that as another deliverable that we're including in our project. You can imagine across all these multimodal data sets, there's a lot of opportunity to do data integration and to do some integrative analytics. I'll give an example of that in just a minute. But this is a big focus of this year and next year, is building out the data integration across these different domains. So just as an example, of the harmonized MRI data that we have so far, about 800 of those participants also have autopsy data available in this data set. So it's an incredibly unique resource and an opportunity to better understand, everything from disease subtyping for the different types of ADRD to imaging signatures of a neuropathology that, we haven't had the sample sizes to look at previously. So really excited to build out some of those data integration projects and make those integrated data sets available to the research community as well. We do have a website. This is actually down right now. I apologize, it's down today for repair. But keep this this code and you can look it up. It should be back online tomorrow. But we have a resource where you can go on and look at what data are available. We have a searchable data dictionary for every phenotype that was included here. You can even go in and see what cognitive test was used across different studies that map to the different PHC scores that we generated, the imaging features that are available, all that's available on there. We have a data dashboard that tells you about, where we are in the process, how many data sets are coming down the pipe, when to expect the next release. And we even have a data explorer tool where you can look at some summary information across age for a given phenotype and look at how memory performance, for example, changes over the course of aging across these different cohort studies. So really excited about this resource, continuing to develop and grow that. Apologize that it's down today and you're not able to look at it, but it'll be back online very soon. And then I mentioned that another huge priority for the project is educating the research community. So we've done that a few different ways. One is partnering with the Alzheimer's Association to do some webinars. Last year, we did a basics of data harmonization webinar in April 23, followed by a pre-conference immersive workshop that was hands-on for anyone who wanted to learn about cognitive data harmonization. We had 50 researchers come and join us for that conference. And we did everything from walk through how you can access the data on NIGADs to actually get into the weeds in the data and learn how we do the cognitive data harmonization, how you can apply these data with longitudinal data analysis in R. Had a lot of fun doing that. So this year, we're building on that and we're doing it again. And oops, sorry about that. So we have a webinar that we finished in March that was on the basics of neuroimaging data harmonization. And then this year's pre-conference immersive workshop at AAIC is focused on neuroimaging data harmonization, in particular T1 harmonization. And again, that's everything from, this is led by Christos Stavotsko's team at the University of Pennsylvania. And they've got everything from a web tool that you can use to upload your images and get a parcellation yourself for your own data, to a really detailed look at how these different statistical harmonization approaches, post-processing work and how you can apply those techniques and approaches in your own data analysis. So excited for that. I think there still is some openings. So if you're interested, you can register it when you register for AAIC. But yeah, looking forward to doing that again this year. So maybe to finish up, I'll just give a few more examples of some of the ways these data have been used so far and why I think it's such an incredible time to be studying Alzheimer's disease. And I'll just give a few highlights from my team, but there's a lot of teams that are using these data in a lot of different ways. So this is just, again, scratching the surface of some of the applications for these harmonized phenotypes. The first thing that we did was just run a GWAS of memory performance out of the gate, kind of proof of concept, hey, this is working, and this is going to be a really effective tool as the data set grows. And you can see on the top left here, this was led by Derek Archer. We had over 129,000 longitudinal visits in the most recent data set that was uploaded, data from more than 26,000 participants. And yeah, it turns out that when you've got disease in your data set, that the genetics of memory performance in late life reflects the genetics of Alzheimer's disease, because Alzheimer's disease is such a contributor to longitudinal changes in memory performance. So this GWAS identified 4 known loci for Alzheimer's disease and was a nice proof of concept that these endophenotypes are very effective in identifying genetic markers that we know are relevant to dementia. What we're excited about is there's a possibility as these data sets grow more where we can start to deconvolve some of the genetic contributors to late life cognitive change and normal aging from those that are contributing to Alzheimer's disease, dementia, and other types of dementia that are present in a lot of these data sets too. So we're excited to be working on that now, and certainly hope to have some additional results for that very soon. I mentioned the sex differences in APOE project that was led by Dr. Logan Newman-Tracy, so I won't mention that again. But on the bottom here, we also ran a sex-specific GWAS of memory performance, executive function performance, and language performance. And actually, I'll give you a little more detail on this right now. So let me talk about this. This was led by a PhD student, Dr. Jackie Eisman, who completed her PhD last year and is now in a postdoc position at Columbia. And one of the things that she did, so in a genetic study, a lot of times we show a Manhattan plot, which is a plot where every dot is a variant across the genome. So on the x-axis is genomic location. On the y-axis is your negative log 10 of your p-value. So that's how statistically significant that particular variant is. This is called a Miami plot, which is a lot like a Manhattan plot, except in Miami, the skyline is right on the water, so you get a reflection. So it's a cute name for a plot. And what we have here on the top is the GWAS signal in women, and on the bottom is the GWAS signal in men. And here I'm showing you our GWAS for language performance. And first, of course, you can see on chromosome 19 in both men and women, you have a really strong signal at APOE. Of course, APOE is a really strong contributor to late life language performance. But what we found was really fascinating is there was a signal in the VRK2 locus that was specific to women. You can see the signals here, and we don't really see much of a signal there. So one of the things that we can do is called a locus zoom. So now we're going to zoom in really tight onto that locus in the genome to look at what we're seeing. So here in the locus zoom, you can see that this particular variant, it's just upstream or downstream of VRK2. It is an EQTL for VRK2. And you can see that the signal is really strong in women, and there's really no signal in men to speak of at all. And we are really fascinated by this finding for a few reasons. This is an unpublished finding that we're currently writing up. One of the reasons we are so interested in this is that this particular gene has been implicated in a whole bunch of language relevant GWASs recently. So there's a language connectivity GWAS that showed a BRK2 effect. And there's also a musical beat synchrony and developmental stuttering GWAS identified them. What's really interesting is a lot of those phenotypes actually have a flipped sex prevalence, where it's more prevalent in men than it is in women in development. So kind of interesting that this signal that we're seeing as female specific in dementia is related to a signal that we're seeing in early life language alterations that might be more male specific. So thinking a lot about this, still digging into it. We're working with a few groups here who are experts in developmental stuttering, Dr. Piper below, and some of these neuropsychiatric traits to think through it more, but really excited about the finding. And I think highlights some of the ways when you get into more granular cognitive data, you can start to uncover not just novel genetic factors, but also genetic factors that make you think a little bit differently about the way disease is progressing in late life and how these different factors contribute to different aspects of cognitive decline that often are just being summarized in either a case control variable or even a diagnostic variable. So really excited for these types of analyses to continue to grow and to continue to learn more and more. There's a lot. I mentioned the integrative analytics. This is a big focus of the work that we do here, trying to think of ways that we can better model how the disease is changing, particularly in the preclinical stages of disease. Here's just a few examples of those. There's a really nice method that came out recently where you can quantify using longitudinal amyloid PET imaging data how long an individual has had amyloid in their brain and track it back to an age of onset of amyloidosis. So we're calculating these phenotypes as well and using these as endophenotypes in genetic analysis. The other thing that we can do is use that longitudinal cognitive data to model a lot better how an individual participant is changing leading up to and following Alzheimer's disease diagnosis. So you can see in this mixed effect sigmoidal model, you've got this normal aging, I guess you could say, trajectory that's happening prior, 10, 15 years prior to the onset of mild cognitive impairment, and then an acceleration that happens at the mild cognitive impairment stage, and then again, another change in the trajectory that happens after an Alzheimer's disease diagnosis stage. So these types of modeling approaches we're really excited about, and these are additional things that we're trying to release in the AASP to layer in new ways of thinking about the disease, but also where the genetic factors are most relevant across both the preclinical and clinical stages of the disease. So within my team, we use these data. We're interested in genomics for the purpose of identifying targets that we can really act on and that we can move towards therapeutic intervention. So all the data that I was just showing kind of sits up here in our discovery framework and part of our discovery platform. We also partner with Dr. Catherine Kazarowski and others who work in genetically diverse mouse models of Alzheimer's disease to try to align some of the things that we're seeing, and then send those down the pipeline to try to validate and move towards high quality targets that have a genetic basis that we can move towards therapeutics. So that's really the goal of my team. I didn't have time to talk about all the ways that we're doing this with molecular data, but would be happy to talk more about that if there are questions. But I'll just stop there. I'll thank first and foremost NIA for their support of this project. I think it's a really important project. I think it's a huge opportunity for the field. We're really grateful for all the support that we've had from program. Thanks for all the harmonization leads for what they've put in. They put in an incredible amount of time and effort and don't get as much credit as they deserve. So I just want to thank those guys for all their work. Big thank you to NIGADS and to the other ADSP investigators, and in particular, Dr. Lisan Wong. We're grateful to be able to work with such a fun team and make these data available. So happy to take questions if you have them. Hopefully I hit enough on the CDEs, you guys can get a sense of what we're doing and how we're building these phenotypes. But yeah, looking forward to having a discussion about it too. Thanks so much.
Wow, thank you so much, Dr. Homan. What an incredibly insightful session. Your talk was pretty thought-provoking, especially with the insights that you shared, which are relevant to how we can address those challenges as well as opportunities, right, that enable data harmonization. So thank you so much. And in fact, we have a few questions from our audience based on your talk. And so right now we will transition over to our Q&A segment, and so that we can answer some of your questions. Now, before we do that, I just wanted to let everyone know that at the end of this lecture, we do have 2 poll questions that we'll launch just to solicit your interest in future topics, and also to learn what you found most interesting from Dr. Homan's talk. And again, thank you, Dr. Homan, for such a great lecture. And so I'm going to first start by asking you, earlier you talked about, some of the challenges that your team has faced with the data harmonization efforts. How have you been able to overcome that? And, what opportunities do you see in the future in terms of this emerging out?
Yeah, great question. Yeah, so there's a lot of challenge. I mean, the biggest challenge, honestly, is data access. That's the #1 challenge. Most of our time on our team is spent doing data use agreements, going through contracts. Studies are very interested in collaborating, and I can't think of a study that we reach out to that was like, no thanks, not interested. Everyone's really interested, but there's a lot of paperwork that has to happen to make this possible, to make sure that data are being shared in a way that aligns with consent of the participants when that data was captured. And luckily, I mean, NIGADS has so much infrastructure set up around this, and they've made it possible for us to do this type of project where we do a data harmonization, everyone can access it. But man, it's a lot of time that goes into data access. And so, yeah, I would say that's the biggest challenge that I'm happy to talk more about. I can probably talk for hours about some of the, the challenges that we've seen on the data access side. But I think there's huge opportunities that come out of this too. I mean, as the data are coming in, I think one huge opportunity is thinking about, okay, we have harmonized data for more than just the participants that have genomics data on NIGADS. We have it for everyone that's in the cohort. Is there a way that we can develop an infrastructure to make sharing those data easier, right? I think that's a huge opportunity. I think there's big opportunities to integrate some of the real world data into what we're doing. So, we mentioned that MVB project just very briefly, but, in that project, we're pulling in clinical images across 4 different VAs in the US and trying to run some of those through our pipelines to see how we do. And I think that kind of is the bottom level of what I think could be a huge opportunity to integrate in more clinical imaging data into what we're doing as well. So those are some of the opportunities, but I think one thing I would emphasize is that the work that we've delivered so far is going to be so much more valuable as more and more data come in. The studies from Africa, for example, we haven't incorporated at all yet. And unfortunately, it just takes a lot of energy and effort to keep up a resource like this because all of them are longitudinal. So for each of the studies I've already shown, in addition to bringing in new studies, we also have to go back every year to the studies that are already in, contact them, pull in the new data that have come, reharmonize, update our harmonization to include those participants, and then upload those too. So it's a lot of energy and effort that kind of to keep this stuff up to date. But I think the payout is massive, and I think the value add is really, really high. So yeah, we're excited to do it. It's been a lot of fun to do it. But yeah, the data access part is a huge challenge that we've had to overcome.
Yeah, I can only imagine, Dr. Holman. I appreciate your insights there. As a part of the Data Access Committee with NIGADS, I know what you mean when you say how important it is to address data access challenges, mitigating that at the lowest level as possible to enable those investigators to actually access the data, analyze the data, right? To be able to share that data back to the researcher community to further analysis, to further engage in the research, right, towards improving human health related to ADRD. And I greatly appreciate you sharing insights. And I know more work is to be done. And kudos, as you mentioned, to Dr. Lee-San Wong and his team, who have been just really great to work with personally over the last year and a half, and devising ways in which we can actually address those challenges in a very robust fashion.
Absolutely.
And so I wanted to ask another question about, well, looks like we're going to turn over directly to the audience. Someone was asking whether or not your slides will become available. And yes, we will not just share the slides, but also the recording from today's lecture. It will be shared with all of the attendees and those who weren't able to join today. We know how eager you are to review those content. So thank you for asking. Let's see, we have a question here about, what is your workflow around, defacing or de-identifying neural imaging data? Speak to that.
Yeah, great question. Yeah, so it's really interesting because that space has changed a lot since we were funded. And right now our protocol is that we go with the protocol from the cohort study. So if the cohort study is defacing their data, then we'll work with defaced data. A lot of the cohort studies are still releasing data that is not defaced. So we're working with those data and again, releasing what aligns with the study's preference. I think what we're starting to see in a lot of the initiatives that are beginning now is that defacing is becoming standard practice for a lot of the cohorts that are coming in. And that infrastructure is present on Loney. So Dr. Arthur Toga oversees defacing for the Alzheimer's Disease Neuroimaging Initiative and for a lot of the clinical trials that are coming in. A lot of the data that we have in ADSP have a project on Loney now as well, whether that's Wisconsin Registry of Aging Prevention or YCAP, HABS HD, all those are up on Loney. So we could implement a defacing protocol that aligns with the ADNI protocol, for example, down the road. So that's something we've talked a lot about. And of course, our imaging experts have a lot of opinions on this too, as you can imagine. The implications for processing are important to think about too. So as of right now, we're not doing a defacing protocol across the board, but we do have the infrastructure to do it, and certainly have thought a lot about whether we should start implementing that. Yep.
Yeah, that's great. And thanks. It's great to know that it's actually in the pipeline. And at some point, we'll learn more about that. It looks like we have another question or a few questions turning back to data access. It looks like we have a member from our audience asking how they can gain access to the NIGADS database. You mentioned earlier some of those challenges that we're currently trying to overcome right now. Could you speak to that very briefly?
Absolutely. Yep. So if you want to access data that I was talking about today, you can go to dss.nigads.org. I popped that into the chat. On that website, you click apply to access data. And the way that we set this up is that if you get access to the ADSP umbrella study that you can click when you're requesting data on NIGADS, you'll get access to everything that I just talked about. That's just part of the umbrella study. There's not a separate process that you have to go through. Just go through one process and you'll have access to everything. You can feel free to reach out to me directly or to Shannon Turner, who's a program manager that oversees the harmonization project. If you have specific questions about harmonized variables or accessing data from the cohort studies that are not included in ADSP. So for example, we harmonize all the data from NAC, regardless of whether or not they're in the sequencing project. when you do a NAC data request on nacdata.org, you can check a box that says, I want the PHC phenotypes, and they will send you the phenotypes that we harmonized as well across domains. That's true for ROS-Map Mars as well. You can request them for ADNI, for ACT, et cetera. So we're happy to help facilitate that as well. If you need to access the phenotypes that are outside of the ADSP. But yeah, all the ADSP stuff, there's no additional steps. You just need to request access to ADSP and you're good to go.
Thank you, Dr. Hellman. And also, you've actually answered quite a few questions regarding data access, as well as not just to the genomic data, but also to the associated phenotypic data. So thank you for that. And as you mentioned, there's a link in the chat, and there are some pretty comprehensive instructions online to provide you with information on how to actually submit a data access request to the NIGAD's data repository and its other related resources. So Dr. Hellman, I know we have a few more minutes. I'll keep going down the list of questions.
Sure.
During your presentation, thank you. During your presentation, you showed the ADSP PHC, or the Phenotype Harmonization Consortium Leadership Team, and how those members represent different domains relevant to data harmonization. For the ADSP project, you mentioned domains that were established based on certain techniques that are used to leverage how that actually comes to be. You also named domains such as neuropathology data, structural MRI, and PET imaging. But you also said that there are some challenges such that there needs to be an agreed upon data harmonization domain strategy. Could you share more about that, and what techniques were used to identify the existing domains that are part of the PHC?
Yeah, absolutely. Yeah, so yeah, we kind of had to scope it out on what we thought was feasible to accomplish. And we had to prioritize, what things should we include or not include. And there's a couple of questions in the chat that I think are great about other things that we didn't include that maybe we could. So out of the gate, the things that we were most concerned about were ADRD relevant biomarkers and neuropath measures. So can we get in as much information we can about whether or not someone is amyloid or tau positive, and whether or not they have other concomitant pathways of injury. So those were the things, that was kind of our framework for what we were thinking about as we were pulling in data. And then from that framework, the next part of the process was, okay, so there's this type of data that are collected to measure things that we think are relevant to ADRD. Is there an agreed upon way that you can actually harmonize those data if they were captured in different ways? And for a lot of all the domains I listed up there, the answer was yes. We felt really confident in the cognitive approach and the approach for a given domain. We were able to reach out to a domain lead who agreed to do it. Good to go. For some of the other ones that we didn't end up including, I'll just give a few examples. We didn't end up including ASL for a measure of blood flow. We didn't end up including FDG PET. We didn't end up including fMRI. We didn't end up including blood-based biomarkers. Those were all things that we talked about. And the reason we didn't include them was some combination of we weren't confident in the harmonization approach that was in the literature, or we couldn't identify a domain lead who could do it, or the time constraint, where we just felt like the amount of work, we wouldn't be able to deliver something in a reasonable amount of time. So those are things that we're still thinking about, like, can we integrate some of these components in? FTG PET, I would love to bring in. I think that was a huge opportunity for just as an example. But yeah, but it comes down to it's kind of a practical consideration, but also, is there enough in the literature to believe that if we do the harmonization that it's valid?
Wow, incredible. Thank you so very much. We're down to our last minute. This has been such a fruitful lecture, and I greatly appreciate your insights. We have actually quite a few more questions from the audience, and I apologize, we won't be able to cover them now, but we will provide email responses to those questions and make sure that we distribute them to all of those who attended and those who weren't with us. But I wanted to end by thanking our esteemed speaker, Dr. Hellman, truly for your insights and for this lecture, and also to our audience for being engaged throughout the lecture with many stimulating questions. And again, Dr. Hellman, thank you very much for providing us with highlights on the importance of computational data harmonization, as well as viable approaches towards achieving this to enable organizations and institutes, some of which you mentioned, with refining and unifying diverse data sets, while also paving a way for clear insights, including machine learning, data quality, and smarter data-driven decision-making. With that, I just wanted to also mention to the audience that we do have 2 poll questions that we launched. We know many of you are excited about Dr. Holman's talk, and we'd love to hear what you thought was most interesting about today's talk, or what other topics you might want to see in the future. With that, we are at the top of the hour. I wanted to again greatly thank our speaker, Dr. Tim Holman, as well as our attendees for joining today's lecture. And again, we will respond to all questions that we did not get a chance to go through during the Q&A segment. Thank you to my colleagues, and thank you so much for your time joining our lecture today. Take care.